Welcome to EPG Pathshala. Hello, I am Veda Krishnan, scientist in the Division of Biochemistry, Indian Agriculture Research Institute, IARA, New Delhi, India. Today we are going to explore on the topic production of enzyme from the paper Protein Biochemistry and Enzymology. This module has been designed with few objectives. They are to understand what is microbial fermentation and what are the various types. What are intracellular and extracellular enzyme and what are the various approaches and steps in enzyme preparation. And finally, how enzymes can be produced at industrial scale. To start, have a look at this concept map which outlines the lecture. Let's first see what is microbial fermentation and what are its types. Next, we will briefly understand about intracellular and extracellular enzyme. With this preliminary information, we get into detailed method for obtaining intracellular or extracellular enzyme preparations. This lecture will also focus on industrial method on enzyme production. Industrial enzyme production has expanded rapidly in both volume and quantity, owing to the expanding market and increasing demand for the novel and stable biocatalysts which have established their potential application in various industrial sectors. Although microorganisms constitute the largest and the most dominating source of enzyme, several enzymes have also been obtained from plant and animal sources. Production of a microbial enzyme initially involves screening of microorganism with desirable and useful property. The level of natural enzyme production from an organism is often very low, and therefore it is essential that the production levels are elevated to meet the industrial demand. This is often achieved by optimization of the medium and culture condition or by causing mutation in the organism. The microorganisms used for enzyme production are grown in bioreactors or fermenters under optimized medium and culture condition. Both solid state and submerged fermentation methods are used industrially. These enzymes are separated from the microbial culture using centrifugation or filtration and then stabilized to maintain suitable shelf life. Fermentation technology involves the growth of microorganisms like fungi, yeast and bacteria under well-defined conditions for production of enzymes which have immense industrial applications. Fermentation can be of two types. As you can see in the first figure, where the production of enzyme by microorganism occur in a liquid nutrient media. This is submerged fermentation. But when the cultivation of microorganism and further fermentation for the production of enzyme if occur on a solid substrate, then it is known as solid state fermentation. This fermentation takes place in the absence or near absence of water. Solid substances like sugarcane bagasse, wheat straw, soybean cake, oil cake, fruit pulp, etc. are used as the source of carbohydrate, protein and other nutrient for the growth of microbes and subsequent enzyme production. So friends, till now we discussed what is solid state fermentation. One of the main impediments in solid state fermentation is the difficulty in controlling the temperature during the process. The heat which is generated during metabolic activity of the microbe growing on solid substrate is not easily dissipated and is an issue of major concern. Increase of temperature beyond a certain level will often lead to denaturation of enzyme and also indirectly affect the growth of microbe, leading to a reduction in the protein yield as well as quality. Submerged fermentation process is preferred over solid state fermentation, mostly because of a better handling and control of the process conditions. Enzyme produced by fermentation can be intracellular or extracellular. What are intracellular enzymes? These are the enzymes which are produced internally within the cell for use inside the cell itself. For example, respiratory enzymes involved in mitochondria of a cell are produced and used intracellularly. So what about extracellular enzymes? These are the enzymes which are produced inside the cell but released externally for biocatalysis outside the cell. For example, the digestive hydrolytic enzyme like proteases, amylase, lipase are released by microbes into the fermentation medium for hydrolyzing large protein, carbohydrate or fat molecules into smaller fragments for easy absorption of the cell. As you can see, figure outline a schematic overview of the various stages of obtaining an enzyme preparation. Post-microbial fermentation as well as the plant or animal tissue disintegration, the primary step is the solid liquid separation, which is required for the initial separation of cell mass from the broth or the removal of cell debris after cell breakage and further collection of the filtrate. This can be achieved by filtration, centrifugation or aqueous biphasic partition. The filtrate so achieved is further processed to obtain the desired enzyme product. Centrifugation separates the liquid and solid phases on the basis of particle size and density. For enzyme that produce extracellularly, the culture broth is centrifuged at very high speed of 10,000 to 12,000 rpm per minute at 4 degrees Celsius to obtain the filtrate, which contains the enzyme of interest. The temperature is kept low during the centrifugation to prevent the enzyme from getting denatured. The microbial pellets so obtained after centrifugation may be further disintegrated to obtain any intracellular enzyme of interest. Generally, centrifugation is the most preferred method for recovering extracellular enzyme. Intracellular enzymes are also used in significant quantities and must be released from cells by cell lysis and processed for further use. Cell lysis involve the disruption of the cell wall and membrane to allow cellular content to come out, which is then centrifuged under high centrifugation speed and cooling condition to prevent denaturation, to remove any cell debris in the form of cell pellet and yield a soluble extract containing the intracellular enzyme. Some types of cells, for example, some gram-negative bacteria or animal cell are readily lysed by gentle treatment such as osmotic shock, while some others, for example, yeast, gram-positive bacteria are able to resist this shearing force and no cell breakage happens. Consequently, a variety of cell disruption techniques have been developed based on the type of the tissue. In the next slide, you can see the figure which gives an overview of various techniques of cell disruption. This figure gives an overview of various types of cell disruption. As we can see, cell disruption method can be broadly understood under two categories: non-mechanical method and mechanical methods. Non-mechanical method involve cell lysis by physical, chemical, enzymatic or by desiccation. Mechanical method involve shearing of the cell using some mechanical force like homogenizer, bead mill and many more. Some of the most commonly used method of cell lysis are discussed in the coming slides. Sonication is a widely used and simple small scale method used commonly for cell disruption. This involves the treatment of microbial cell in suspension with high frequency ultrasound wave to disrupt cell wall and membrane, which result in their disruption. As you can see in the figure, The sinusoidal movement of the mechanic probe that is immersed within the culture suspension caused by ultrasonication lead to perforations or cavitation within the cell. However, a lot of heat is generated during cell sonication, which may often lead to damage of sensitive cellular biomolecules. Also, ultrasonication leads to the production of very fine debris, which can hinder further processing of the sample in the coming downstream steps. This method can be used as a continuous lysis method on laboratory scale. However, the large scale use of ultrasonication is often avoided due to high operating cost. High pressure homogenizers are another well accepted technique for cell disruption, readily adapted to large scale and continuous operation. The principle of this technique lies in application of high pressure to microbial cells. The microbial cells are thus subjected to impact or shear, and a severe pressure drop across the valve tend to disrupt the cells as the pressure is released abruptly. These shear forces produced are not capable of damaging enzyme released in the solution, and hence it is very safe. This method has been used to break microorganisms like Aspergillus niger, Escherichia coli and Bacillus megatherium. This figure shows the basic component of a high pressure homogenizer. Another method for mechanical disruption of microbial cell is the use of bead mills, which you will see in the coming slide. In this case, small steel, alumina or glass or silica beads, usually a size of 0.2 to 1 mm diameter, are used to agitate cell suspension, during which they get broken or disrupted due to the high liquid shear and collision with the beads. On an industrial scale, high-speed bead mills accompanied with cooling jackets are used to agitate the cell suspension. The grinding chamber is filled with almost 80% bead and a shaft with impellers fixed within the chamber, which rotate at high speed generating shear forces. The efficiency of cell disruption in a bead mill depend upon the amount and size of the bead, the concentration of the cells in suspension, as well as the speed of rotation of the agitator. The bead mills are inexpensive and can be operated continuously on a large scale to lyse algae, bacteria and fungi. Mechanical disruption method suffer from several drawbacks. Because cells are broken completely, all intracellular materials are released. Therefore, the product of interest must be separated from a complex mixture of protein, nucleic acid and cell wall fragment. The nucleic acid so released along with intracellular content may increase the viscosity of the solution and may complicate subsequent processing step, especially filtration and chromatography. The cell debris produced by mechanical lysis often consists of small cell fragment, making the solution difficult to clarify further. Additionally, the complete release of intracellular component often require more than one attempt through the disruption device, adding to the problem by further reducing the size of the fragment. These are the difficult to remove by continuous centrifugation. Furthermore, mechanical method expose the cell to a very harsh condition, and most enzyme will tend to get denatured by the heat generated in the instrument. The non-mechanical approaches to cell disruption are discussed in the next few slides. These can be studied under three broad categories: physical method, chemical method and enzymatic cell lysis method. Under physical method, osmotic shock is a cheap, gentle and convenient method of releasing enzyme, but has not apparently been used on a large scale. Changes in solute concentration around a cell cause a rapid change in the movement of water across the cell membrane. When concentration of the solute inside the cell increases as compared to that of the outside cell, water enters the cell in large amount in order to establish osmotic balance, causing it to swell and burst. Use of this approach to cause cell lysis for releasing intracellular enzyme should indeed be cheap, gentle and a convenient method. However, this method has been not much used on a large scale. Another method is the breaking down of cell by subjecting it to heat. This refers to the use of chemicals to solubilize the component of the cell wall to release the internal component of the cell. However, it is required that internal cellular component including enzyme are sufficiently robust, are insensitive to the chemicals being used and should be easily separated from the chemicals after lysis. So, what are these chemicals? First is alkali, like NaOH, which can be used for the extraction of some bacterial proteins. Other than alkali, organic solvents are also used, like methanol, ethanol, isopropanol and butanol, which can also be used to disrupt the cells. Next is detergent. Detergents can also be used to lyse cells. Detergents such as Tritonex 100 are commonly used for releasing enzyme bound onto cellular membrane, but these are often difficult to be removed from the final product. Sometimes detergents like trimethyl ammonium bromide, which is a cationic detergent, or sodium lauryl sulfite, which is an anionic detergent, are used for cell lysis and can even denature cellular protein, and therefore their removal after cell disruption is a must. Cell disruption using enzymatic lysis method offers a gentler approach of releasing enzyme under milder condition where the enzyme is not subjected to heat or shear. Enzymatic lysis is widely used in the laboratory scale but is less used for industrial purposes. Lysozyme, sourced from the hen egg white, is often used to lyse the beta-1,4 glycosidic bond present in the mucopeptide of the bacterial walls. Gram-positive bacteria are more susceptible to enzymatic lysis than gram-negative bacteria. The major drawback to the use of lysozyme on a large scale is its cost, yet hen egg white lysozyme is the only lytic enzyme available commercially as of now. Cell lysis approaches often damages internally released cellular component as shown in table 1. Cell lysis using mechanical and non-mechanical method often lead to generation of heat, shear stress, release of proteases, foaming and metal toxicity. Interference by detergent or other chemical which may interfere with enzyme and destabilize or inactivate it. The various interfering parameters, their effects and possible solution are shown in table. Please go through. The lysate which is obtained after cell lysis is further clarified. The extract which is obtained after cell lysis is clarified, and clarification refers to the removal of any particular matter remaining cellular debris or other suspended contaminant in the extract which are not compatible with chromatography or any prior to downstream processing. The clarified sample of soluble protein thus form the starting material for further purification assays. The production of microbial enzyme through large-scale fermentation technology account for a significant proportion of the enzyme being produced by biotechnology industries. Fermentation is usually carried out in large bioreactor. The enzyme producing microorganism of choice is allowed to grow in closed fermenting bioreactor with rich and continuous supply of nutrient containing and oxygen. Microorganisms break down these nutrients for their growth and subsequently release the desired enzyme into the solution. You can see this in this figure. In industrial processes, batch fed and continuous fermentation process are mostly common. What is batch fed process? In the batch fed process, the reactor is fed with fresh sterilized medium and the fermentation takes place. When the fermentation has finished, the content of the bioreactor are emptied, cleaned and then again refilled with another round of fresh medium, which is further inoculated and the fermentation process starts afresh again. Thus, fermentation is said to occur in batches. What is continuous process? In the continuous process, the sterilized medium is introduced into the fermenter at the same flow rate as the fermentation broth is leaving the system. This leads to achievement of a steady state production. In other words, the microbial cell continuously receive fresh medium, product and base product are continuously removed. Continuous reactors can be many times more productive than batch reactor because of the following factor. The continuous process fermentation allows easy control and optimization of the bacterial growth rate. The reactor can be operated for long period of time without having to be shut down. To summarize, dear students, we have gained an understanding on enzyme production, approaches and methods. In detail, we discussed about submerged and solid state method of microbial fermentation. We have also seen about various steps which are involved in an overall enzyme production process, as well as a method for obtaining extracellular and intracellular enzyme preparation. In addition, the fermentation in bioreactor for large scale enzyme production in continuous and batch process have also been studied. After these enzymes are produced, they are harvested from the fermentation medium and clarified. Post clarification, the enzymes are then concentrated and then subjected to purification procedures and used for a plethora of applications. Thank you.